Hi, it's Alissa speaking. So how is cell division regulated? Well, cell division is regulated by protein complexes that contain cyclin-dependent kinases, CDKs, which are enzymes that turn different processes in the cell cycle on and off. The lines in the graph represent the different CDKs that are made during the cell cycle. Kinases are often found abundantly inside growing cells, but don't do anything because they are inactive. In order for the kinases to become active, they must be attached to a cyclin and is therefore known as a CDK. So this would be a CDK. CDKs act as internal signals and cause the cell to move through the stages of the cell cycle. However, CDKs do not allow the cell to pass through this cycle automatically, as there are still checkpoints that the cell must pass through first. Internal and external signals provide stop-and-go signs at each cell cycle checkpoint. As mentioned previously, CDKs are internal signals. MPF, the mitosis promoting factor, is a complex that contains cyclin A and B and is attached to a CDK, meaning when CDK is activated, it will interact with different proteins and allow the cell to proceed to the mitotic phase or M phase. External signals can influence cell division through a growth factor. The growth factor is a protein released by specific cells that stimulate other cells to divide. For example, the platelet-derived growth factor, or PDGF, is required for the division of cells in connective tissues. Without PDGF present, the cells will fail to divide and move through the cell cycle. The picture shows what a petri dish of cells with and without PDGF looks like, and it is clear that cell division doesn't occur without PDGF. A disruption in the cell cycle can result in many problems, but one that we are all familiar with is cancer. Cancer cells are usually a result of a disruption in the cell cycle control. As mentioned before, the cell cycle is very regulated, so a cancerous cell is often found to have mutation in cell cycle genes that disrupt the proper cell cycle. Here you can see a normal DNA strand, but when I add the colors, which represent the mutations, this becomes a mutated DNA strand, which will not respond normally in the cell cycle. Cancer cells will exhibit the ability to continuously divide despite overcrowding or detachment from the tissue, as shown here. So this is a normal cell and this is a cancerous cell. Also, the cells may not need growth factors to grow or divide, as they will make their own growth factor or convey a growth factor signal that is not present in the cell. During the cell cycle, cancer cells stop at random points, unlike normal cells, which stop at the G1, G2, and M checkpoints, which is shown in this diagram. So instead of stopping at the 3 normal checkpoints, they can stop anywhere along the cell cycle. Basically, this means that cancer cells have abnormal cell cycle control system due to mutations in the cell cycle genes. So that is the cell cycle control system, and I hope that is helpful.